Alright, so we are going to start off with our questions around free energy. So as I mentioned, I've reorganized the order of the questions. That way they kind of flow and make a little bit more sense. So I'm starting off with question number 1, which is, can you explain how the relationship between surface area to volume affects biological systems and the efficiency of cells? So we talked about this a little bit in class, but let's say you have these 3 cubes. And one is a 1 by 1 by 1, the next one is a 2 by 2 by 2, and then finally 3 by 3 by 3. So what we're seeing in this is that our highest surface area to volume ratio is going to be in the first cube. On the handout that I gave you guys, that's how you're going to calculate out surface area and volume, and remember that these formulas are both given to you on the AP test as well. So I attempted to do this with the potatoes, and it worked a little bit, but we'll have to wait until we get our agar cubes to get a little bit of a better dye here. But essentially what they did in this experiment was they placed all 3 of these cubes in whatever liquid they were using to do their experiment in at the same time, and left it, my guess was probably about 15 minutes. That's usually what you're going to do. And what you can see is that in the first cube, where's my mouse? Here it is. In the first cube, we had the color go through, looks like all the way through. Here we have it come through maybe about a centimeter, and finally about a centimeter again. We can see it was about a centimeter for all of them. But because the surface area to volume ratio was so high in this first one, it was able to penetrate the entire cube. Here it was able to penetrate almost all of it. You can see that there's some in the middle that didn't get it. But in this large cube, we can see that there's a big area that did not get any of the dye, and that's because the surface area to volume ratio is the smallest in this cube here. So how does this affect efficiency of cells? I answered that one kind of on this next question too, because we're talking about biological processes and efficiency of cells here. So as you can see in our cubes, here's a 1 centimeter cube. Our total surface area for that one is going to be 6 centimeters. Our volume is 1, our ratio is 6. Now if we go up to that larger cube, so this would be like that last cube that we saw in the example before, we can see our surface area to volume ratio is very small. So let's say that we were trying to have oxygen or nutrients, anything that we need to get into the cell in order to do any sort of biological process. Well, in a large cell like this, it would take a really long time for those things, whatever they are, the materials, nutrients, matter, to diffuse through the cell. So that's why we have so many little cells, because then we have a really high surface area to volume ratio. So essentially for both of these questions, 1 and 11, having a high surface area to volume ratio will speed up the rate of biological processes. Okay, the next one is question 12. Can you describe how organisms utilize matter to grow and reproduce? And question 16, which is, can you describe how organisms utilize energy to grow and reproduce? So living things have to utilize both energy and matter in order to grow and reproduce. So what we're looking at here is this word metabolism. Metabolism is what can transform that matter and the energy that an organism receives into whatever else they need it to be. So it's the totality of an organism's chemical reactions that they do in order to survive. Some of the major ones that we're going to be looking at is photosynthesis and cellular respiration. So I want to look over here at these equations. Our first one is photosynthesis here. And I think that this does a really good representation showing how important matter and energy are to our systems, because here we can see that energy is utilized in order to take carbon dioxide and water and turn it into C6H12O6 and oxygen here. So we can see that both matter and energy is transformed in order to give us our products. But we can also take it the opposite way with cellular respiration. We can take those molecules, we can take this matter, and we can transform it into, once again, different types of matter and energy. So number 19 said, can you explain how energy moves through and affects biological systems? So first of all, what even is energy? Energy is the capacity to do work. All energy can be categorized into two main forms, which are kinetic and potential energy. Kinetic energy is the energy of movement, and potential energy is basically any stored energy. So we're going to look at potential energy a ton in biology because this is our chemical bonds. So for example, if we look at cellular respiration, we have potential energy stored in our bonds here of our molecules that is, once they're broken down, is released. Now it's important to know that there's two main metabolic pathways. There's catabolic and anabolic pathways. So catabolic pathways break down complex molecules into simpler ones, and they release energy. So looking at cellular respiration and photosynthesis, which one releases energy? That would be cellular respiration. We're breaking down complex molecules into simpler ones. So cellular respiration is an example of a catabolic pathway, whereas an anabolic pathway builds up more complex molecules, and it needs energy in order to do this. So photosynthesis is an example of an anabolic pathway. Now, really important to know when we talk about energy are two of our laws of thermodynamics. These are the two that we're going to be discussing the most. That's why I have those only. Our first law of thermodynamics states that energy is conserved. Energy is not never created nor destroyed. It's just simply changing form. And living organisms have the capability to take part in changing its form. So for example, we had that potential energy here, and we were able to release that. Our second law of thermodynamics states that spontaneous changes that do not require outside energy increase the entropy. Entropy means disorder of the universe. So make sure to write those two down. Okay, question number 15 is, can you explain how free energy changes affect organisms, populations, and ecosystems? So when we talk about free energy, I want you to start thinking of Gibbs free energy. Now that I think about it, okay, I wrote it here. Gibbs free energy, write that down. Okay, I'll go back. A living system's free energy is energy that can do work under cellular conditions. It's important to know that living systems will increase the entropy. Once again, entropy is disorder of the universe. Now what we'll do is we'll use our change. We know it's a change because we have this triangle. The triangle is a delta. So whenever we see a delta, that means change. So change in free energy, which is represented by the G. This change in free energy tells us whether or not a reaction is occurring spontaneously. So is it requiring energy in order to do this reaction? Oh my gosh, this ball just ran by me. Or not. So if a reaction has a delta G that is less than 0, that is a favorable reaction, so that would be spontaneous. An unfavorable reaction, so non-spontaneous, has a delta G that is greater than 0. So let's go ahead and do one of these sample problems. You'll be given a lot of the information, so the nice thing about our Gibbs free energy problems is that you can just kind of plug them in. So here is our first sample. Calculate the Gibbs free energy change for the following chemical reaction. So ATP to ADP plus phosphate. So, and this is super common in our unit right now, looking at photosynthesis and cellular respiration. So the reaction occurs at 68 degrees Fahrenheit. The change in heat, delta H, is 19,070 calories, and the change in entropy, which is represented by this delta S, is 90 calories per Kelvin. So if we look at this, we know that we have our temperature in two different units, so that's going to be the first thing that we're going to need to change. So once again, we're calculating for Gibbs free energy, which is represented here by the delta G. This is our change in heat, delta H. T is temperature, provided right here. And delta S is change in entropy. So we need to start off by making our temperature in the same units. We are always going to be using Kelvin in this. So in order to get to Kelvin, we take our 68 and it becomes 293. I will give you guys a resource in class that has all of the ways to calculate the different temperature units. Unfortunately, that is not provided for you guys on the test, so that one is something we will have to memorize. Okay, so now we have our temperature in the proper units. So delta G is 19,070 calories. We got that from here. Minus 293 Kelvin, that was our temperature, times delta S, which is our entropy, which is 90 calories per Kelvin. So we're going to go ahead and calculate that out. I'll give you a second here. Okay, so our correct answer is a negative 7,300 calories. Now we always are going to give our answers in kcal. So complete your equation doing calories, and then at the very end transfer it over to kcal by moving your decimal place 1, 2, 3 to the left. Now it is so important that we make sure we know if this is going to be negative or positive, because that's going to be what tells us if our problem is, if our free energy change was spontaneous or non-spontaneous. So going back, looking at this, favorable reactions or spontaneous reactions is delta G less than 0 or greater than 0. If our answer is negative 7.3, is our reaction spontaneous? Yes, it is, because our answer is negative. Now what that also tells us is if this reaction is endergonic or exergonic, and we know that it's exergonic because the answer is negative, energy has been released. Now if our answer was positive here, that would be not spontaneous, and that would be endergonic. Okay, can you compare and contrast endothermy and ectothermy? This is problem number 18. Endotherms are able to maintain their body temperature and are not affected too much by the external environment, so for example humans and birds, whereas ectotherms rely on the external environment in order to maintain body temperature, so it's going to fluctuate a lot. An example of this is snakes and most fish. Okay, question number 17. Can you compare and contrast heterotrophic and autotrophic nutrition? So once again, it's really important to look at all of these roots here, because that is going to give us huge hints. You should see that already. We talked about endergonic and exergonic. It's important to know those. Here we are again, hetero and autotrophic. So auto is going to be self, trophos is going to be feed. So that tells us that autotrophs are able to basically make their own food. So this would be anything that's conducting photosynthesis and chemosynthesis. Sorry, I don't know if you can hear that. It is a squirrel in the tree. Where you're able to convert light energy or chemical energy into food. Heterotrophs are required to take energy through an outside source. They do not have the capability to turn sunlight or chemicals into food, so that's why we eat things. So we are considered heterotrophs. Okay, and I think that is our last question then for free energy and matter, which is the first part, our first kind of major category of our questions. I'm going to move into cellular respiration and then our photosynthesis questions in a different video. Thanks for that.